Dr. Jim Holland, who's also a reserve geneticist. That your
That's right.
title also with USDA ARS at North Carolina State. And you'll be talking to us about maize genetic diversity, a resource for crop improvement.
Right. Thanks a lot.
Yours.
So, yeah, I'll shift from soybeans to corn for a little bit and tell you about some of what I'm doing. But of course, today the focus of today is on ponds. So what I want to tell you is how some of the things I learned as an undergraduate student working in his lab in 1988 and 1989. Some of these things have stayed with me, and I mean, I think I've gone pretty far in life. I'm pretty amazed how far I've gotten with it. So let's talk a little bit about corn, and at each point I'll try to tie this in with some of the things I learned 20 years ago here. So corn is an extremely diverse crop, both genetically and phenotypically. Here's phenotypically meaning how these ears of maize look. These are different collections from around Latin America. At the genetic level, at the sequence level, any two inbred lines of maize are as different from each other as humans and chimpanzees are. Maize is about 10 times higher in sequence variation than humans. And humans are actually pretty diverse, so there's tremendous diversity. Most of the diversity, however, is restricted to tropical Latin America. And here's an example. There's a paper I did with some colleagues in Mexico where we looked at the 59 races of maize and how they were distributed across ecological zones in Mexico. There's tremendous ecological diversity for where corn is grown. In Mexico, you have corn being grown on mountainsides and places where it would never be grown in the U.S. And that's been going on for probably 10,000 years. And so there's been adaptation and evolution for these very specific uses and ecological adaptations. In contrast, essentially 99% of the U.S. maize is represented by a single race, which is the corn belt dense. And that's the kind of corn we're used to seeing. Another way to put this is if you go into the central plateau of Mexico and just go through one village and collect ears of maize, you will find more genetic diversity in that one village than in the entire U.S. corn belt. However, U.S. maize is very productive, and one reason for that has been breeding. So here's a typical pedigree chart. These are all released commercial. These are private company inbred lines, many of which, for example, derive from the Iowa State inbred B73. But the point of this slide is that there's things like B73, these key sort of founder ancestral lines get used over and over and over again. And what this does is it really narrows the genetic base of what we're working with. So we know that corn belt dents are extremely productive, and that's good. We have high-yielding corn in the U.S., but still we believe that there's all this diversity still available in tropical maize that probably could contribute to increases in productivity. We've actually shown that that's the case. And also we know, for example, there's plenty of good disease resistances. And now that we start thinking about using corn for different types of food purposes, tropical germplasm has a lot of unique grain characteristics that are simply totally absent from the corn belt dents. And just in general, the idea is diversity is a good thing. If we can enhance the diversity of the U.S. corn crop, this should provide buffering against any emerging new pathogen, either species or strains or populations. The problem is, it's not so easy to just grab some tropical material and cross it onto U.S. temperate maize. A major problem is photoperiod sensitivity. So tropical maize is by and large very sensitive to long days. So if you grow them here in our summers, we have long day lengths. They grow very, very, very tall. So it's probably sort of nice for forage, but they often don't flower. They'll just keep growing vegetatively until the day lengths get down. This is actually an inbred line. Inbred lines tend to be very short, but here's my quite tall former grad students standing up showing this inbred line from, this is actually Thailand. And you can see there's actually no ear that's produced on this, and the tassel's kind of gone crazy because it's been under long day lengths. So we would like to access this germplasm, these genetic resources from tropical Latin America, but in order to do that, we need to have some approaches where we can deal with some of these issues of bad adaptation, let's say. And I'll talk a little bit about some of our genomics approaches, and this follows very closely along with what Dr. Matthews was talking about. We want to know, how do we relate DNA sequence variation to the trait variation that we're interested in, so we can actually identify the genes involved in conferring these differences. And this is what I like to call phenomena. So what I'm really interested in, not just DNA variation, I want to know what's going on at the phenotypic level, at the level of the traits that we really want to manipulate for breeding. And then, of course, the other approach is conventional breeding, and actually that's been very successful. My colleague, Major Goodman at North Carolina State, has been doing this for a long time. It actually is very effective. It does have to be long-term. Actually, I think this approach is also going to be fairly long-term, but it is effective. And I'll talk a little bit about some of the things that I'm doing with that. So, what does this have to do with Devine? Well, one of the things, if we get into the genomic stuff I'll talk about, is this is all really based on linkage analysis. This goes back exactly following along with what Dr. Matthews was talking about. If you establish linkages between marker genes and genes controlling the traits of interest, those can then be manipulated for breeding. And the basis of this is completely based on linkage analysis. And I learned linkage analysis over here in building 011A or something like that. And this is the little red book from Mather that Tom came to me and he said, "You need to read this book to understand linkage mapping." And I did. I actually, before I went to grad school, I had actually plowed through much of this book, and that's gotten me a long way. So if we had an army of 5 billion people reading Mather's little red book, we'd all be in good shape. And so just to follow up, so when I was working here in '88 and '89, a big focus of this program was linkage mapping, and he really laid a lot of the groundwork for the modern soybean genetic maps by establishing linkages between many of the Mendelian variants. And so this is just like a figure from a paper he wrote. These are, he had to go through and set up all these crosses. And, it was very careful planning, setting up these crosses, and then identifying where you have linkages between genes. And here's a couple marker genes that do something. I'm not a soybean guy, I don't know, but that establishes, this is linkage group 16, and then you can overlay the DNA markers on top of that. Okay, so linkage analysis is useful for identifying genes that have strong effects, like soybean cyst nematodes, but now we also want to use them for analyzing what are the genes involved in complex traits, things that are controlled by lots of genes and have environmental influences. Now we need to shift to DNA markers because these supply an abundance of mappable genes and they permit whole genome coverage. And the first time I ever did DNA extractions and ran markers was in Dr. Matthews' lab while I was here. And then you can, again, then though it's basically linkage mapping. And that's the basis for what we call QTL mapping. And QTL mapping is simply trying to identify those genes that affect quantitative traits. And the difference is now you have multiple genes, each of which might have a smaller effect, but combined they could have large effects. So I'm just going to give you two quick examples of what we've done pretty recently in maize. And the highlight of both of these is what I'm calling joint analysis of multiple populations. One of the problems up till now, people have been doing QTL mapping for 15 years or so. But a lot of times what happens is you make a cross, you identify some genes that are affecting the trait, and then you go to a different cross, maybe the breeding cross you actually want to apply the information to, and it's actually totally different sets of genes segregating there. This phenomenon is called genetic heterogeneity. The human geneticists have a nightmare of a time with this. It turns out for many human diseases, different families actually have different genes that control those things. So there's no like diagnostic markers for these. It gets very complicated. So what we want to do is right off the bat, instead of just taking one population and then later doing a second population, we'll set up a series of crosses to make multiple populations right away. And so we did a project where we crossed a couple of temperate lines. These are US inbred lines to two different unrelated tropical lines. So we made 4 F1 hybrids. Each one has one temperate parent and one tropical parent. We self-pollinate for 7 to 8 generations to develop recombinant inbred lines. These are just inbred line progeny. They're not the direct children, but you can think of them as the children of each of these crosses. And each of them carries a pair of chromosomes where they've got recombinations between the two parental allele combinations of each of these. And then here you have to make a linkage map. So in this population, we made a linkage map with 1,340 markers. So we've got markers of almost 1% or more, pretty dense. And then we, of course, had to evaluate these in the field, and that took a long time. But eventually what I want to show you here is, this is the kind of result you can get. This is chromosome 10. This is the map of chromosome 10. We've actually got markers all the way through here. I didn't draw all the markers. But what this graph is, this is a log plot. You see these a lot in these QTL talks. This is just basically, what's the statistical significance that there's a gene at that position affecting that trait. So the trait here is silking date, which is female flowering, response to photoperiod. And you can see that these lines down here, these are from, if you map each of the populations individually, these are pretty high lot scores, so there's pretty good statistical evidence. If you combine the information together across populations, you get into like world record lot scores. These are things that human geneticists would kill each other to get. Okay? And we can get them in plants because we can set up these populations and grow them all under common environments. The other thing that's very nice is these little bars here. That's an indication of the resolution. How confident are you of the position of this thing? In the joint analysis, we get it down to a centimorgan for a QTL, and again, that's very good. So we're now dealing with one centimorgan, which is 1 out of 1,500 centimorgans in the whole genome. So now we can narrow down and we can ask the questions: what are the genes in this region, and can we figure out what they do? The other thing we've learned is that when you do this, you find out that yes, indeed, different parents carry different. The alleles those parents carry have different effects. And so you get these complex segregations depending on the way the crosses go. Okay, so the second example of how we've used linkage analysis is this nested association mapping population in maize. And the idea here was, okay, that was nice, we had 4 populations. Let's really try to sample as much of the genetic diversity of maize inbred as possible. So here's a phylogeny of, these are just random maize inbreds selected from all over the world, and they group into, these 2 groups are sort of the temperate groups. There's stiff stock and non-stiff stock. That's, corn breeders all know what these things mean. But here's the tropical stuff down here. And then you have some weird outliers like sweet corns and popcorn. So what we've done is we've chosen B73 as a founder line. And the reason is B73 is the key ancestor of modern U.S. maize. It's also the reference sequence for the maize sequencing project. So this sequence is nearly complete. And then we went along this graph and sampled 26 other lines that we could use to cross to B73 to generate segregating populations where we're now accessing genetic diversity all across this huge phylogeny of maize. And these are what the ears look like. I mean, that's a little bit hard to see. Okay? So here's the way the crosses are made. We sample, these are the different 26 founder lines. Each one gets crossed to B73 like this. So you cross, say, B73 to CML52, you make an F1. And then out of each of these, we've generated 200 recombinant inbred lines. That was a lot of work. In the end, we've made 5,000 inbred lines, new mapping lines. This is obviously not something I did, on the side. This is a major project. Sorry, I got the monitor thing on. With Ed Buckler at Cornell. He's USDA at Cornell, and Mike McMullen, USDA at Missouri, and Steve Kresovich at Cornell. And again, here's just another way to think about it. You sample all this diversity, cross everybody to B73, generate these lines, and now we've developed a linkage map up here. That has 1,100 SNP markers, so it's quite dense. Then you have to phenotype this thing, so there's 5,000 lines you have to deal with. It's really a lot of work. But anyway, we've done it. And there's just a tremendous amount of diversity. These are random pictures I've shot walking through the field. Here's all these different tassel types. Race root variation is tremendous. And ear types, you have long skinny ears and sort of short stubby ears. So any trait you want to measure, there's a huge amount of variation in this population. Then you can associate that phenotypic variation with the linkage map, and that's what this thing is. This is another genome scan. This is the whole genome now. And again, it's just this blip, 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 blip. These things just mean, okay, in this region there's a QTL, in this region there's a QTL. Again, the log scores are huge because we've got this joint analysis of 25 populations.